Welcome to Pharma Stability Series. If you want to understand stability studies in a simple, practical, and regulatory-focused way, you are in the right place. In this video, we will break down a key stability topic step by step, including its scientific basis, regulatory importance, and common industry expectations. So let's get started.
All right, let's talk about something we all rely on, our medicines. We just trust that they're going to work. But what really happens when they fail over time? The easy answer, the one most people think of, is that it's just a simple chemical breakdown. But today, we're going to dig into the real story, and you'll see it's about a whole lot more than just chemistry. It's about system failures hiding in plain sight. Let's get into it.
So let's start with that big question. When you hear that a medicine's stability has failed, what's the first thing that pops into your head? For most of us, it's probably one single, pretty straightforward idea.
And that's the common story, right? It's the tip of the iceberg. The chemistry just goes bad. The active ingredient breaks down. Simple, but that's not the real story. The real story, the massive part of the iceberg hiding under the water, is that long before the chemistry ever gives out, the system designed to watch over it has already started to fail. A stability failure is almost always a symptom of a much, much deeper problem.
And you know what? Those failures often start right at the very beginning. We're talking about cracks in the foundation, problems that are literally baked into the study before a single sample ever gets pulled from the shelf.
It all begins with a document called the Stability Protocol. Now think of this thing as the constitution for the entire study. It is the official blueprint. It lays out every single rule, what you're going to test, when you're going to test it, and what passing or failing actually looks like. If this blueprint is flawed, the whole house is going to come tumbling down.
So what does a weak blueprint look like? Well, it's full of holes. Maybe the storage conditions are unclear, or the dates for testing, what we call pull points, are kind of vague. Maybe the pass-fail criteria are ambiguous. Each one of these little holes creates confusion, it opens the door for mistakes, and it pretty much dooms the study right from the get-go.
And here is another huge crack in the foundation, the problem of the perfect batch. It's so tempting to pick a sample that was made under ideal, pristine lab conditions. But think about it. The whole point of the study is to prove the product can survive out in the messy, real world with all its normal variations. A perfect batch gives you data that looks amazing on paper, but shatters the second it's put under any real-world pressure. Okay, so even if you manage to build a solid foundation, things can still go wrong while the study's actually running. Let's dive into the errors that happen in real time.
How many mistakes do you think it takes to cause a serious problem? 10? 5? Nope, it takes just one. Missing a single scheduled sample pull. It might seem like a tiny slip-up, right? But what it does is it rips a hole in the data's story. It creates this gap in our understanding and immediately makes you question the credibility of the entire study.
See, a missed pull isn't just about a missing data point. It's a massive red flag. It's a symptom that signals all these deeper issues you see here. Poor planning, weak oversight, and just a fundamental breakdown in discipline. It's proof that the system itself isn't really under control.
I get this. Sometimes the failure has nothing to do with the drug itself. You can have a perfectly stable, rock-solid formula, but if you put it in a package that doesn't protect it from light or moisture or oxygen, the product is still going to fail. And let's be crystal clear, that is absolutely a stability failure.
All right, this next part is maybe the most subtle, and honestly, the most dangerous part of all. These are the failures that are hiding in plain sight, buried inside data that, if you just glance at it, looks perfectly fine.
This quote right here, it absolutely nails it. Just because a single test result is technically passing the test, it does not mean everything is okay. The truly crucial thing isn't one data point. It's the story that all the data points tell you over time. This is where teams can completely miss the flashing red lights.
This table is the perfect example. Just look at the impurity level. At 6, 12, and 18 months, every result is below the spec of 1.0%. So technically, check, 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 it's passing. But hang on, look closer at the trend. It goes from 0.2% to 0.4% to 0.6%. It is literally doubling every 6 months. This product isn't stable. It is on a clear, predictable path to failure. Ignoring a trend like this is like ignoring smoke because you can't see the flames. Yet.
And there are other silent traps too. Things like weak data review, where teams just miss those trends we just talked about. And then there's documentation.
You know, in the world of pharmaceuticals, the rule is brutally simple. If it wasn't documented, it didn't happen. You can have a perfectly executed study that gets thrown out because of incomplete or sloppy records.
So, when we put all of these pieces together, the flawed foundation, the real-time mistakes, the hidden data traps, a really powerful final lesson starts to emerge. This isn't just about one little lab test.
It's about a system. A successful stability program is a team sport. It's a relay race. It starts with development, designing the product, then it gets handed off to manufacturing to produce it, then to QC to test it, it's overseen by QA, and finally it's scrutinized by regulatory. If any single person in that chain drops the baton, the whole system is at risk of falling apart.
And that leads us right here to the harsh truth. A failed study is almost never just a science problem. It is a failure of alignment. It's what happens when those three critical pillars, the science, the systems, and the discipline, are not all in sync.
Which leaves us with this final and I think most important question. We have to reframe how we think about this. The real question isn't, is the product stable? The much, much more important question is the system stable? Because at the end of the day, a robust, disciplined system is the only thing that truly guarantees the medicine you count on will be safe and effective every single time.
If this helped, like and subscribe.